Hello, everyone, and welcome to today's webinar titled DNA Synthesis for mRNA Therapeutics, How to Ensure Good Results by Choosing the Right DNA. This webinar is a part of the 11th annual event in the Genetics Week virtual event series. I am Sydney McNeil of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher. For more information about Thermo Fisher Scientific, go to www.thermofisher.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. In addition to utilizing the live chat feature, we encourage you to participate in 3 poll questions throughout the speaker presentation. Click on your answer when the poll questions are displayed. If you have any trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. I now present today's speaker, Axel Trefzer, Director of Product Management at Thermo Fisher Scientific. For a complete biography on our speakers, please visit the Presenter tab from the menu on the left of your screen. Axel, you may now begin your presentation.
I'll be speaking about DNA synthesis for mRNA therapeutics and how to ensure good results by choosing the right DNA. Today, I'll cover a number of different areas. First, I'll give a brief introduction on who we are at Thermo Fisher and what we do. And then I'll talk about synthetic biology and where it comes from and some of the background by explaining how this brings engineering to biology. And then I'll talk about one of the secrets in synthetic biology, synthetic DNA, and in the end, how this really applies to mRNA therapeutics. So, Thermo Fisher, who we are the industry leader in serving science. We are a pretty large company with 80,000 employees and roughly $30 billion of annual revenue. There's a large commitment in the organization to innovation and science, so more than $1 billion are invested. A large group of well over 5,000 scientists and engineers are focused on development and research. I think the goal of what we do is really lead by developing leading innovative technologies and to provide deep application expertise to our customers and scientists in the field to move science forward. On the next slide, you'll see some examples on how we are set up. Basically, it's centered around 4 large areas: life science solutions, specialty diagnostics, analytical instruments, and lab products and services. And the way to look at this is, lab products and services are a lot of the things you use every day in the lab. Analytical instruments are the big boxes providing you with in-depth analysis and specific applications in a number of areas. Specialty diagnostics is a lot about diagnosing pathogens and other items, immune markers and so on, biomarkers. And in the Life Science Solutions group, it's a lot around different reagents, instruments, workflows in life science research centered around recombinant DNA, proteins, antibodies, and all of those things. We have a mission we are very proud of, which is we enable our customers to make the world healthier, cleaner, and safer. And here a couple examples are given. So one example, how we make the world safer, is really to provide, in this case, instrumentation to ensure the quality and safety of medicines. For cleaner, we support innovative research and development of cleaner, more efficient power sources. And healthier, we play a significant role in both delivering diagnostic tests as well as vaccine and therapy development and production to fight the COVID-19 pandemic globally. And this is really, as I said, a mission we're proud of, and also something that makes me go to work every day. And a key aspect is really here, we enable our customers. So we really want to make it easier for you to do your research.
As a reminder, we would appreciate your participation by answering a few polling questions. To record your answers, please click on the option on the screen, and for list options, select all that apply. Our first polling question is, are you interested in getting a quote on a protein expression project for your future research? I will give you a few moments to select your answer. All right, Axel, I'll go ahead and turn it back over to you.
With that, I want to move into more background on synthetic biology and what this really means in terms of, bringing engineering to biology. When we look at biological research, one key aspect that's really changed a lot of things over the last couple of decades is sequencing data and the exponential growth sequencing data has provided. It follows Moore's law and really has accelerated our understanding of all kinds of life on this planet, whether it's plants, animals, microbes, what have you. And this in-depth understanding has really enabled us to move biology forward from a science that's mostly centered around observation to a science that's more driven by application. And this deep understanding of the biological code at the center has really driven a lot of that transition. When we look at this, synthetic biology is a step in the evolution of biological research. And, humans, ever since we've used biological systems and we've aimed at developing biological systems with desirable traits that started out with, classical breeding. And when you look at that, it takes a long time, centuries, millennia to develop organisms which have the desired trait. Over the last century, accelerated breeding was used, which accelerated that whole process down to decades. For the last 30, 40 years, genetic engineering has played a predominant role which again accelerated the way how we can produce biological systems with desired phenotypes, and also has enabled us to generate phenotypes and genotypes which go above and beyond what nature really provides. And the way we look at synthetic biology, it's the next step of the evolution of biological methods, how to generate desirable phenotypes. And it really accelerates this by bringing a different mindset to biology. And what I mean with this can also be explained by this statement of synthetic biology is trying to make biology easier to engineer. And this engineering approach in biology is a key aspect. And this, when you look at it, can also be exemplified by this classical engineering cycle of design, build, test. When you think of designing a biological solution, it really means that you have to take your vision and you have to translate it into a biological code, which in the end means you have to translate it into DNA. And this is also where we see our role. We've provided DNA synthesis services for the last 20 years. And the key aspect in this context is really, you have bio-IT solutions which enable you to design your biological code that encodes your vision. And then you need tools that enable you to efficiently write that DNA, and this is what we provide to our customers in the market. Ultimately, once you've encoded your vision in the right DNA, you've embedded it in the right organism, you're able to then basically follow this engineering cycle. You can build your prototype, you can test it, you can redesign it, and ultimately you will have a system that does what you want and has the phenotype you want. While this may sound futuristic, it is actually not that futuristic anymore. I think it's something that's over the last couple of years has impacted many areas in life sciences and also beyond. Some of them are shown here, whether you talk about, drug development, agriculture, enable new biotechnology, like, the whole discussion around bio-based fuels, bio-based chemicals. Vaccine research is something that's really also being driven by SynBio. Obviously, a lot of what we do in any basic research can be applied in any basic research field. And last but not least, also synthetic DNA and synthetic biology and writing DNA has also enabled new applications beyond life sciences like DNA for data storage.
Okay, our next polling question is, what is your primary source for the DNA you need for your experiments? Option 1, isolate, amplify, and clone it myself. Option 2, buy ready-made clones. Option 3, custom gene synthesis service. Option 4, I don't use DNA in my experiments. I will give you a few moments to select your answers. All right, Axel, I'll turn it back over to you.
With that, I want to move into talking some more about, synthetic DNA and the secrets behind it. Synthetic DNA comes in a number of different flavors and forms. It can be, linear, it can be cloned, and it's basically a ready-to-use plasmid. It can be provided in a functional vector that can go right into the application, or it can also be just linear fragment that just encodes the DNA and then either has to be cloned or can be used as a linear fragment. We also provide additional services like larger scale plasmid production or direct production of protein from your target DNA, or in some cases, for directed evolution purposes, provide pools of DNA which encode a certain amount of diversity, which enables screening of the right genotype or phenotype. The underlying product and how it's made is shown on the next slide. In the end, anything we do around DNA synthesis really starts with the customer and the researchers on your end, who uses a design tool, bioinformatics, to design the right piece of DNA in the right construct. It's transmitted electronically, it's ordered. We then produce it using custom synthesis and then supply the final product to you. All of the detail in the middle is shown off to the right here. We again start with bioinformatics, then it's oligosynthesis. We assemble, clone it, we confirm that it's correct by sequencing, provide the documentation, then we ship it to you. This workflow is again shown in some more detail on here. What I find important is really in the beginning, bioinformatics does serve two purposes. One is it enables us to design a sequence in a way that we can reliably and efficiently build it in the minimal amount of time. At the same time, it also enables us or customers to design a sequence that's fit for purpose and the biological function that you envisage, so you can actually optimize your sequence for your function. The next step, oligosynthesis, is really traditional chemical oligosynthesis. So we produce short single-stranded oligonucleotides, which then in the gene assembly step get assembled into double-stranded fragments, which is then cloned into a vector for amplification and clonal isolation in bacterial systems, and then sequencing to confirm the correct sequence and that it matches whatever scientists and customers ordered in the front end. We invest significant effort to continuously drive efficiency and improve our services, accelerate the service, and make it more seamless for our customers. So one large area where we focus is really to drive throughput and by miniaturization and automation. Sequencing is actually a significant bottleneck in the workflow, so employing high throughput sequencing, NGS sequencing, really increases the service level and accelerates the process. And in the end, what this does, it allows us to deliver up to 12,000 constructs, up to 12,000 base pairs in as little as 5 business days. And as I said earlier, if you need more than just your custom sequence, we can obviously also provide it in your target vector and application vector. So it's ready to use and enables you to think about your application and your science and focus on that. And don't worry and don't deal with the tedious aspect of molecular cloning and handling DNA anymore. As I said, bioinformatics plays a pivotal role in this. And not only for us, because it's critical for us to design the sequences correctly for production, but also how to ensure that you get a sequence with the best biological function. And I'll explain this some more in the next couple slides. So one aspect is to really ensure that you get the most out of your DNA. And what that often means is that you need to really ensure that you get a high level of expression or the ideal level of expression of your target sequence. And so we've developed a methodology which we call Gene Optimizer, which in mammalian systems and other systems provides you with an opportunity to optimize for high protein expression. In this context, we take into account over 20 parameters, including codon usage, GC content, cryptic splice sites, known instability sequences, RNA, secondary structure, repeats, and so on and so forth. By designing your sequence correctly, we can ensure that you get the highest output possible. And I'll show some data on the next slide exemplifying this. I'll walk you through this in some more detail. So what we did here was take a set of human proteins, express them in a human cell line. We took a wide range of different functions representing highly expressed proteins such as transcription factors, ribosomal proteins, low to medium expressed proteins, certain membrane proteins, which have different folding kinetics, and also proteins or peptides, which are actually typically not expressed at all, like cytokines. And what you can see in the Western plot is always in triplicates, wild type versus optimized. And what you can see in all of these examples, and this is a subset of what's published, there's a reference at the bottom. You can see that in all of those cases, we achieved a significant improvement of expression. And I want to emphasize, we took human sequences and expressed them in the human cell line. And we've seen this over and over again, also when we take heterologous sequences, that using our approach can really ensure that you get good expression of your target sequence. This obviously shows you how you can express biological function. There are obviously, with that we also looked at how can we look at adding additional functionality to your DNA, so not just the biological function. And that's something I want to explain a bit more in the next 2 slides. So when you look at digital media, it's actually possible to easily watermark a document. And what watermarking really means, you embed a message into the media and the document, which is not visible to the user at a first glance. And this is exemplified here using a digital image. And if you take a small subset, and this is a grayscale image, just for simplicity purposes, you can see that each of the grayscales comes with a number. And if I now want to encode, I can take a simple ASCII code, which consists of 8 bits. And I can then basically look at, if I want to encode a certain message, I can slightly modify this grayscale, the grayscale of different pixels, which then enables me to, in this image, encode a message by slightly altering the coloring. But in the end, when I look at the complete image, I won't see that change. So this enables me to encode a message which is only visible if I know a message is there, and I also know how the message was encoded, and then I can go in and check for that message, decode it, read it, and understand what it means. We were wondering whether similar approaches can be applied to DNA, and what we did there was also is shown on the next slide, to look at how can we actually encode it in the coding region. So in the protein coding region, because that's typically the message or the part of the protein of a recombinant DNA construct that is required for its biological function. And the approach we took there was to look at, what's the layer of information which we can adjust without actually significantly altering the biological function. And that's where we then started looking at the genetic code and its redundancy. And when you look at the bottom right, you can see a subset of the human code and usage table. And there's a number of amino acids which are encoded by 4 or more synonymous codons. And what we did there is that we actually looked at the rank and used the rank ordering to then encode a bit. And when you go to the top, you can then also basically, by using these amino acids, step through the coding region, and by slightly altering the coding region without affecting the biological function of the complete construct and the complete sequence, we can add a 3rd layer of information as a watermark and encode additional information in a heterologous construct.
Okay, that brings us to our last and final polling question for today. Would you like to speak with a technical sales specialist from Thermo Fisher about options for gene synthesis and protein expression services? I will give you a moment to select your answer. All right, and thank you so much again to all of those that participated in our polling questions today. Axel, you may go ahead and continue your presentation.
You may wonder what this can be used for. It's actually something that can be used in a variety of ways. So you could add, a brand label or label vaccines or gene therapies. You can incorporate information on ownership, origin, manufacturing date. And what it really does, it ensures that your open reading frame or your target sequence can be traced without any doubt where it goes, and you can always really go back and verify that this is the sequence that originated in your lab and in your design. In addition, it also will allow you to clearly distinguish a synthetic sequence from any possible wild-type source. If you look at optimized sequences differ from natural sequences. So, even just optimization will actually do that, but you can also ensure that you take out any doubt on that. And in our view, in the long run, methods like this can also guide us towards setting standards for how do we actually label recombinant sequences and GMOs in a universal way. With that background on what's writing DNA and DNA synthesis, and especially the aspects around DNA design, I now want to move into how this applies to mRNA therapeutics. When we look at mRNA therapeutics, we offer a wide range of different aspects and products that support this workflow. Whether it's all of the, in the center, all of the reagents that go directly into production of the actual mRNA, or whether it's downstream, anything that it's needed for purification or formulation, or upstream when we talk about DNA synthesis services to design your target sequence, which is used as input for the mRNA production, or whether you talk about plasmid production, vector prep to produce it. I want to again center around DNA synthesis, obviously, because that's the title and the purpose of the talk, to explain how that plays a role in mRNA therapeutics. And when we talk about this, it is really important to understand that how DNA synthesis plays a role in mRNA therapeutics is very much tied to the application. And obviously, when we look at, what do mRNA therapeutics do, I think we all know this and we all understand it. mRNA therapeutics are a way to reprogram cells for diverse applications. Whether this is, you basically transfect the mRNA and then you produce some sort of protein in the cells, whether this protein becomes an antigen, which then allows you to basically use mRNA as a vaccine, whether you produce enzymes or cytokines, which then can overcome certain limitations in the body, whether you use it to express Cas9 or guide RNA to change the genotype of a cell, or CAR T approaches. All of this can be achieved by mRNA as a modality, and I think there's many more ways to come in the future. The point I want to make is that depending on your application, the approach and how you supply the product is different, which then ultimately also in terms of what you need as input material at the DNA level is really different. And I want to use two examples from the vaccine space to illustrate that. One, if we look at something like COVID, which obviously has driven a lot of interest in mRNA therapeutics over the last year. In the end, it's really, we deliver one in theory, we deliver one template DNA and one sequence to basically 8 billion people globally. And when you look at this in terms of, what does it mean for the input DNA, the plasmid DNA, the DNA synthesis that happens, you need to synthesize one template DNA, and then you need to provide hundreds of kilograms of DNA that can be used as input for the mRNA. And also, the DNA can be produced on stock. So there you basically have the challenge of how do you scale it to that massive scale that you can provide mRNA and you can provide the input material globally to everyone who needs it. When we look at the other end of the spectrum, I want to use cancer neoantigen immunotherapy as an example. It really starts with a tumor biopsy, which is sequenced, and then it's being analyzed to identify which mutations are present on the tumor. How does this allow you to select epitopes which allow the immune system to distinguish the tumor from healthy cells? And then these neoantigens need to be synthesized and efficiently delivered to patients either via plasmid DNA, viral vectors, or mRNA. And then, the challenge is very different. In this particular case, typically this is a personalized approach, or often this is a personalized approach. So it's one treatment for one patient. Also, it becomes very time sensitive. So, the synthesis portion needs to happen ideally in significantly less than 2 weeks' time to enable that, you can go from biopsy to treatment for the patient in a very short period of time to make sure that this happens before, that this happens in time for the patient to benefit from the treatment. And this represents very different challenges on how to provide the DNA construct, which I'll explain some more in the next slide. When we look at how we actually do DNA synthesis in the RUO space, which I explained a little bit earlier, we actually do this in a very sequential way. We start with design, and then we move into oligosynthesis, assembly, cloning, do some QC, we scale it, and we do final QC, and then we ship out to our customer. If anything goes wrong at any point in time, we do step back, we repeat, we either step back one step or multiple steps, or even in some cases have to go back. What that means is that, and this is shown by the blue line, that within the time window which we offer to our customers for our services, we do not achieve 100% reliability. However, when you look at personalized therapeutics, especially the example I was just giving on cancer immunotherapy, you obviously want to ensure that in the promised time you as a customer or researcher receive 100% of your constructs. So how do we get there, and also how do we actually achieve this in a time that's significantly shorter than the typical time which we have for our RUO constructs? That's shown on the next slide. I mean, in the end, it's pretty straightforward. We look at it differently from a design perspective. We really make sure that we design that in the upfront, bioinformatics analysis, we really identify all of the potential shortfalls and risks, and we address them as early as possible. In addition to just our standard synthesis approach, we actually right away start with a backup option. We also, as we move through the process, as much as possible, run parallel options, not just, we take one approach, we look at how it works, and if necessary, we step back. We actually often run parallel processes with slightly different protocols or complementary approaches to ensure that in each step we achieve 100% first time right, to ensure that as we step through it, everything is achieved in the shortest time possible. In some cases, we do parallel processing. Another point I want to make also clarify is that when you move into therapeutic applications, typically additional specifications apply compared to typical cloning and research use only. So as early as possible, we also make sure that we address these additional specs. We also provide the options for extended QC. And in the end, we also ensure that the whole process is documented. Our products at the moment are all ISO 9001 certified. But again, with the extended documentation, we have the option to provide products which are qualified to go into further downstream processes that can go into mRNA therapeutics and other therapeutic applications. And in the end, this approach ensures that in the shortest amount possible, we can deliver a highly qualified product that is well documented and also at a very high reliability, which meets the needs for therapeutic approaches as well. With that, as I said, we can guarantee shortest turnaround time from order to shipping. We have utmost order fulfillment and high reliability of 100%. And also, I want to again point out that the optional optimization we provide can deliver you the highest in vivo performance possible. Also, we provide the option to provide additional genetic labeling / branding, which allows you to really trace all the way through the whole process. Did anything change? Did anyone actually steal my sequence? And again, as I said, we can actually provide customer-specific requirements and any sort of special needs around documentation. Whatever is possible within the confines of our current processes is something we can provide. And again, we have flexibility in our manufacturing scale and can go up to high microgram / milligram quantities of DNA and provide DNA that's ready for downstream processing in GMP production. And with that, I want to summarize. I think, bringing engineering to biology allows us to design the best functional molecule for all biological applications. The secrets of synthetic DNA really provide us with control over these complex processes and enabling new approaches, and also ensuring that you, as a researcher and scientist, get the best possible DNA for your science. And this also enables us to tailor this to support mRNA therapeutics and embed specific technologies in the process to ensure that in the end you have the best possible outcome also in this space of mRNA therapeutics.
Thank you again, Axel Trefzer, for your time today and for your important research. Before we go, I would like to thank the audience for joining us today and for their interesting questions. I would also like to thank our sponsor, Thermo Fisher, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until May 18th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.